Thank you so much, Chair. Um, my name is Abel Ramuelo from University of Pretoria, from the Center of Environmental Studies. Today I will be talking to you on a topic, rangeland assessments and monitoring in the ever-changing environmental condition. I'll be really looking at the Earth observation and geoinformation science applications. The presentation outline really will take, um, you know, the conventional outline, just looking at mainly the global environmental changes, national perspectives, remote sensing as a tool of monitoring the objectives of the talk, trends in the use of Sentinel-1 and Sentinel-2, nutrients estimation using remote sensing, biomass estimation, and also looking at the long-term vegetation changes. And I also then look at the tree cover mapping and also invasive species mapping. And then I'll provide just few bullet points on the summary and what I see as the future. Okay, maybe before I start, let me try to explain in few words the center that I'm involved in. From University of Pretoria is the Center for Environmental Studies, that is CIFES. It is housed in the Department of Geography, Geoinformatics, and Methodology. CIFES offers a course-based master's as well as research-based master's and doctoral studies, and is also pursuing interdisciplinary research in the field of environmental sciences, looking at the host of research areas. What are the rangelands actually? Rangelands are really this vast lands of our globe, covering higher proportions of our land. And the rangelands really provide variety of ecosystem services, grazing and browsing resources for livestock production, which is very important for the livelihood. So I'm sure we're going to be hearing a lot about this. And then also provide, like biodiversity, the issues of species populations, and then also habitats, and then to some extent, in most cases, play a crucial role in carbon sequestration. On the latest report in the EPBES and IPCC report, they really spoke about, these three nexus, the biodiversity, the climate change, and the good quality of life, and then how they're interlinked. And then today we see human activities really influencing the biodiversity and also climate. Yes, if you look at the major drivers um, of the state of our rangelands today, and then also the state of the biodiversity, we find this report, the EPBES and the IPCC, are trying to sort of highlight the key direct drivers and then also the indirect drivers. And then all this, and most of those, drivers are mainly due to human activities. For example, the invasive species, direct exploitation of resources, and there's also pollution, and then land conversions as well. And then also others drivers are more indirect, and then all this lead to biodiversity decline, and they also exacerbate climate change, which in turn affect the quality of life. Then, if you look at the climate projections, and it is clear that the future is not looking good. The future is showing us that the average temperatures are likely going to be increasing in future. and then that will also exacerbate, the frequencies of drought. We have seen of late that droughts have been very frequent in most part of our country, and that has a lot of implications on our economy and then also on our rangelands. I think when it comes to precipitation as well, it is clear that rainfall is going to be declining in future, and then, or rather, is going to be more erratic, also linked to the drought as well. And then there's a latest study now by Scono et al. That we're also looking at the transformation that our ecosystems are experiencing in the country. And it was clear that, South Africa also lost about 0.12% of the natural vegetation per year since 1990 and 2018. And then if you look at the rate of loss between 2014 and 2018, it was highly accelerated as compared, to 1990 and 2018. And this shows that there's really transformative changes that are taking place. And this could be attributed to number one, settlement expansion, and then number two, also looking at agricultural expansions. Those are the key, not excluding others, key transformation that could be taking place. You can read in detail in the paper as published. And then, if you look at all these challenges that are taking place, we see the need for protection and the need for restoration, and also the constant monitoring of our ecosystem. When we look at the restoration, we see now we're entering into the new UN decade on ecosystem restoration. We need to continue to reduce the impact of climate change and also the land use and land cover changes. At least the planning has to be better to minimize all this transformation that we are experiencing. And really, there's a need for monitoring over longer time scales. Using long-term in situ data, it could be field data collected across the country, to satellite observations, which are always available. And then what is very important is to start, developing framework of ensuring that this sort of data set is used for monitoring purposes, and then also for us to start tracking our restoration interventions, and then for planning and management of our ecosystem, even ensuring that we control the number of animals that our land can carry in a specific point in time. When it comes to the monitoring side of things, remote sensing is one of the tools that can be used. Remote sensing has been used now for quite some decades, trying to observe the ecosystems, and acquiring that information, highly needed information, over time. And that is the advantage. One of the advantages of remote sensing data is that it is acquired non-stop, consistently for any part of the world. And then what is simply required is for us to start interrogating this data for our own benefit. There are merits of using remote sensing for monitoring. I've mentioned on my first statement that, of course, remote sensing is not an end in itself, but also a means to an end. Why I'm saying that? Because we still require a lot of field data to calibrate remote sensing data to be able to give us the products that we want. But notwithstanding, there's still a lot of merits of using, remote sensing, like the greater spatial representation of the area monitored, data obtained with greater frequency, coverage of larger or wider areas. We can actually move from the scale of district, municipality to province, all the way to the national scale. And then we acquire this data in a non-intrusive and a systematic manner, of course, using the satellites roaming around, and then acquire information in those areas where those inaccessible areas. But with remote sensing, we are able to do that even in rugged terrains, and then in deep oceans, and so forth. And then there is a possibility of reducing cost, especially when you're developing monitoring programs. And there's a greater speed in providing information to decision makers. The objective of this talk actually is to really showcase some of the results that were sort of derived from various remote sensing studies to assess and monitor what I call the rangeland essential variables. Mainly looking at the quality, the quantity, long-term vegetation changes, and then also maybe looking at the species. And I must say that the species composition, richness, and diversity of the grass, I don't have the result for this at this point, but we've got recently completed studies on that. The pathway of analyzing such a wealth of data, especially if you start looking at the data from the 70s to date, you'll realize that you tend to have petabytes of data sets, including maybe some time series in situ data and other ancillary and critical data that you need to sort of estimate all these variables of interest. We are in the era where now we cannot be complaining about the storage and the data analysis capabilities of our computers, because if you look at it now, with South Africa, for example, we've got one of the fastest computers. Whether most of people have access to those computers, that's another discussion for a day, but for another day. But when you start looking at integrating all these data sets into the modeling environment, especially now using machine learning and all those tools, we are able to sort of extract those meaningful information that we require from the image, be it the quality, be it the quantity, and to some extent start looking at the species mapping. And then from there, with the field data, of course, we need it a lot to ensure that we look at the accuracy of the products that we are generating. This is an example from one of my PhD students who was trying to look at, the use of Sentinel-1 and Sentinel-2 imagery for retrieval of biophysical and biochemical parameters. I mean, this was very promising. You start to see that there's a spike of usages of this data because they're freely available. They can be used to estimate parameters that sometimes Landsat cannot do because of the capability of their red edge, band. I just want to give you an example now on nutrients estimation. And then also LAI using remote sensing. These are one of the key variables that can assist us to understand the biochemical and biophysical part of our rangeland systems. This is an example of using a commercial satellite data since we've used a lot of work, we did a lot of work on using hyperspectral imagery, which is also often commercial because it's airborne. People need to come and fly specific areas and so forth. But when we started, investigating a lot the red edge band that is critical to characterize the biochemical nature of the plants, we had to even purchase a commercial data from WorldView 2. It's a very expensive data. It's about $40 or more per square kilometer. But we really wanted to investigate this red edge, and the results were very promising. Here on the left hand side, you see in the map, covering a bit of Kruger National Park, Sabicens, which is the private game reserve, and also the communal areas. This is around March, which is more towards the end of the wet season, and then on the other side you tend to have the July, which is also just towards the end of the dry season as well. Then you can see that using this data, we're able to characterize leaf nitrogen concentration, and even during the dry season, you can see that most of the nitrogen is along the riparian zones, which could be also associated to, some of the tree canopies that are also picked up in the map as well. Because in this case, it was not really corrected, but most of what you see is the grass. And then the other study is when we started using now the spectroscopy data to try to understand how best we can simulate the Sentinel-2 data and then be able to estimate nitrogen over time without having to extract reflectance from the images, without having to waste a lot of time trying to link the pixel to to the field data. But this way we develop independent models from spectroscopy where we get like canopy reflectance and nutrients data for different grass species over time, put it together, then simulate the Sentinel-2, then be able to go to the map and invert those models to estimate nitrogen concentration. And that has been working very well. And for me, that is the future when we start looking at how best we can develop monitoring systems for estimating nitrogen. You can see the patterns during the wet season and then also the patterns during the dry season. That's why you see that the variability there seems to be very limited, because I used a similar scale just to show the depth of the differences. Yes, our latest work now with Dr. Tsele is to start looking at chlorophyll concentration, because as much as we would want to do nitrogen concentration, often nitrogen concentration samples are very expensive to analyze as well. But there has been a lot of improvement on the models for chlorophyll concentration than the nitrogen. Then the idea is that if we are to characterize chlorophyll concentration very well, we can develop empirical model between the chlorophyll concentration to that of nitrogen concentration. Then we're able to estimate nitrogen concentration using variety of models, not only empirical models, but we can also go to the realm of using physical based models, where we simulate the reflectance as a function of those key vegetation parameters that define the behavior of the reflectance and then so forth. This is an example in Golden Gate. We managed to estimate chlorophyll concentration, and then you can see red showing lower areas of chlorophyll concentration, and then yellowish to green showing maybe moderate to higher values. And then this can also give you an indication of where more animals are found. And then we did the same in Marrakech National Park as well, in the waterback. You can see also the green areas showing where you have higher chlorophyll concentration, and the reddish ones showing a lower nitrogen concentration. And again, the models have been very interesting for both parks. I mean, over they're explaining over 60, between 60 to 70% of the variation of the chlorophyll, which is not bad. Now what you're seeing here is the leaf area index, or LAI, for Golden Gate Highlands National Park. From Sentinel-2, you can imagine now we're starting to look at 10-meter resolution, which is a freely available data. We don't pay anything. We just have to get the data and analyze it related to our ground observations. And then you can now see the patterns, in areas where you have very lower productivity, which is lower LAI, and an area where you have higher productivity, like areas with more than your LAI of 7. The same in Marrakech, where we're able to estimate leaf area index. You can see also the patterns that are showing that the models are able to pick it up, and then again the performance of the model were relatively good as compared to any other type of modeling beyond Sentinel-2. Again, here we're looking at 10-meter spatial resolution data. Is really highly detailed data that can be used further into monitoring or development of carrying capacity models and so forth, especially when it's scaled to biomass as well. Chlorophyll concentration can be scaled to nitrogen, protein content, while the LAI can be then used as a proxy or as a predictor of biomass. This is where I was saying that we use the chlorophyll and leaf area index to try to create models for estimating grass nitrogen, and then you can see that the models are not bad. These are really very interesting models, and the best one explaining over 70% of variation of grass nitrogen. I mean, it shows that there's a potential in this, but of course this needs to be done for different grass species, for different seasons, and then for different sites, so that we can capture a full variability of the relationship. And then when we then develop our remote sensing models, we are easily going to use this, especially when we have limited funds to analyze nitrogen concentration. The biomass estimation side of things, using remote sensing, this is one of the most progressed products, especially when you're using remote sensing to to derive it. Yeah, here we'll talk about biomass estimation based on the Sentinel-1 and the Sentinel-2 dataset through the stepwise multiple linear regression, just a classical method. We wanted to see which variables are going to be coming out significant in explaining the biomass. And then you can see the list, the leaf area index being one of the highest predictor of biomass, and that is not surprising, as I indicated previously, that this is a key variable that really give us an idea of the productivity of the land, of the vegetation. And then you start to see also the red edge type of bands coming out, because they minimize saturation problems when you start estimating biomass. Those are the classical challenges that we have in remote sensing. But here we try to look at the optical part of remote sensing and also the SAR part of remote sensing. When we look at the SAR, which is more the active part, we didn't find any significant model, but on the optical side, we did on the Sentinel-2. But when we combine the two, those are the 8 variables that came out of 18 remote sensing variables. The other example actually is when you estimate, biomass using Sentinel-1 and Sentinel-2, but now using the machine learning techniques, such as random forest, and then you then see, how critical, these data sets are, and then LAI again comes out to be the top, and then we didn't pick any SAR, only, bands as well, and then I think some of the experts of SAR were mentioning that during peak productivity is the time where maybe the optical part of the remote sensing should be able to help us estimate biomass. But when the vegetation starts senescing, then the SAR then will pick up. In other words, then we need to find specific periods when to use which data set. Then that way we constantly tend to have the biomass data as in when is required, because with Sentinel-1 and Sentinel-2, every 5 days you get the data. And then it will be good now to start looking at the time series data, and then how that assists us in estimation of the biomass. Here we tested as well on our latest study, that this was just a conference paper that one of our MSc students wrote, and then here we tend to see again. We just wanted to test as well the Landsat 8, which is one of the good data sets that we have now on the Landsat series. Then we tested the before and after fire type of sampling, and then these are the results you get, and it is really promising. And then these results were surprisingly comparable to that of Sentinel-2 as well, and then which shows that we tend to have more data sets that are very good in estimation of the biomass. Then if we want to start looking at carrying capacity models and so forth, we can start going to as much detail as even 10 to 30, between 10 to 30 meters spatial resolution. This is just an example of biomass map from the WorldView 2 as well, for Kruger National Park and the surroundings. And then you can see that with a high resolution data set, you tend to even identify the smaller patches of those biomass in the areas. And then this data is very critical if you now want to start understanding at a local scale how animals are moving. And then if we could start now having wider view of this nutrient concentration based on Sentinel, then we're able to do even more work. I think now the future is no longer WorldView 2. This was just a demonstration of getting sub meters or less than 2 meters spatial resolution products. But Sentinel-2 can do all that we need. Then another part of the remote sensing result that I wanted to demonstrate and share with you is looking at the long-term vegetation changes. I just got the results for 2 to 3 parks just to show you how useful this data is in terms of understanding, maybe measuring productivity since 2000 and how it is changing. and be able to track, the trends of whether the vegetation condition is increasing or decreasing. You can see here, you've got the actual NDVI variability, you've got the 6 months average, and then also the 12 months average, just to show you generally what is the direction of the of change. And the other things that we can do is to use series of time series analysis tools to look at the anomalies. And then one of the example that I use often to assist, especially in monitoring the smaller parks within sun parks, is to use the vegetation condition index. But here we just use the absolute anomaly. Then we can compare absolute anomaly, relative anomaly, and then also things like your vegetation condition index that sort of quantify the impact of the impact of drought and also grazing on the vegetation. Another example is for Maragele. You can see it's a relatively stable system. And then the other thing is that here we're looking at the mean for the entire park. What is very critical would be to start looking at the mean per vegetation type, so that we understand which vegetation types are more affected than the other. But that information can also be discerned from the maps themselves when we start looking at how the NDVI has been changing for only 1 month over time from 2000. Then this is what you get, just comparing the March month from today backwards, and then that gives you an idea of which areas are improving in terms of the vegetation condition, and the red is those areas that are below normal condition and maybe towards critical anomaly. Then you can do as well the September month, where that is towards the end of dry season before the rains. Then you compare it with long-term data set. Then generally it could be indicating that there is a decrease in precipitation or any other factors that are driving this proliferation of the red. And another one is the Golden Gate one as well, where you tend to then do the same, follow the same process, and be able to understand which areas of the park are experiencing, the below normal conditions or above normal conditions, where the green, is showing you above normal condition, and the reddish is below normal conditions. But you can see most of the park is looking good, and then anything that is less than 50% here shows below normal condition, and anything more than 50% of the VCI, vegetation condition index, tells you about the positive anomaly, which is above normal condition. We did the same for mountain zebra, which is a bit more different from the other parks in terms of the variability of the greenness. Of course, that could be related to the amount of rainfall experienced in the environment. And then from there, you are also able to create the maps and trying to understand which areas are more stressed than the others, which areas are also affected by the drought than the others. This information can give us that. And the other part, maybe the series of the last part of the demonstration of the products that we can, we can generate is the tree species mapping. I just took an example of some of the work we did at Kruger National Park some few years ago, where we were able to start looking at detecting individual tree species using a very high resolution data, I think just about a meter. Then we're able to detect the canopies of the trees and be able to create the maps. Like the top one here is your marula tree map, and the second one is your Spirostachys species as well. That is mainly found in the riparian zones. And then you can have the Acacia nigrensis widespread and the Combretum species as well. Then we're able to, understand, the type of species, and then if we overlay the elephant impact and other data sets, we're able to see which trees are being targeted by the animals. And this is another study where we look at testing the worldview 2, which is a very high resolution image, as I mentioned. It's the same where we have the biomass, we have the nitrogen, but here we wanted to look at the canopies of the trees, and we're able to distinguish between acacia species, combretum, and then and marula and so forth with relatively acceptable accuracy. You can see over 80% of the overall accuracy. And then here we are also trying to use the biophysical and biochemical variables that we can estimate accurately these days to distinguish between your invasive species and then your native species. This is your native species based on the chlorophyll concentration. You can see that they are having less as compared to the invasive species, and the invasive or alien species here I think is Acacia mense, and the other acacia species. And then here you've got, according to LAI, you've got the native species, and then also you've got the invasive species with the higher LAI values. It's very distinct in terms of the biochemical and biophysical constituents. Then what it tells us is that we can use that as a basis to separate these species on the maps. And then again here we also try to look at how best we can automate the process of detecting Acacia mense and other species. And the red is showing you all the invasive species somewhere in KwaZulu-Natal around Harrismith. And then you have the green being native species. Then you can see that we are able to detect those. And then again here we're trying to look at what is the optimum time window for mapping these invasive species. And you can see that around July you tend to distinguish a lot between the Acacia mense and Acacia delbata, which is one of the species. I know that Acacia has been changed to another name, but yeah, you'll bear with me today. We just use an Acacia, but these maps already were showing that we are able to map those species. Now, since we have the cloud systems, we are able to implement this into the cloud system and sort of map wider areas. But of course, more calibration data will be required to do that and to achieve that. And then this is an example. I just wanted to complete with this example, which we saw is our 2018 paper, where we started looking at how best we can use the time series data to estimate the tree cover based on the NDVI, and then look at how the tree cover is changing. And we are able to show areas of higher tree cover, and then areas of decreasing tree cover, and the areas of no change. If you see the black color there at the bottom of the map, you can see that those are the areas where you are likely going to have bush encroachment using almost 20 years of data sets. You can read more on some of the drivers that we were picking up In some rural areas, we've realized that abandoned farmlands are the ones that are showing a positive signal, which is the positive increase in the tree cover. And then because when they are abandoned, then they are down colonized, by the Dicostracis cinerea species or any other sickle bush like type of species. And then you can read more about this wonderful work that we did. Yeah, in summary, our rangelands are changing and we are also losing the biodiversity. Then I think it is time for us to start preventing and reversing and then also restoring our ecosystems. I know there are a lot of initiatives in the country on this. Then we just have to continue to do it. And now, when it comes to the monitoring side of things, tracking the progress of restoration, looking at levels of degradation, mapping levels of degradation, we are at that level where we can use remote sensing tools to do that because the world is far ahead. Most of the people are far ahead in different part of the world. Then we need to start operationalizing this system for our benefits because global products that we can use are of course globally parameterized and might not be used at a local scale. Then local scale decisions will require locally parameterized data. Then we should also capitalize on, the cloud-based systems that are available for access and analysis of the data, especially when you're looking at the Google Earth engines and climate engines that gives us both remote sensing and then also climate data. And then if we integrate all this, you realize that it's possible now to put this products together into a monitoring system or decision support tools that can be used to safeguard our rangelands, as we know that they are very critical and they provide essential ecosystem services. When you look at the future, now there's no need really of downloading a lot of satellite data, storing them in a specific place. It's better we use these data cloud systems where we are able to analyze the data there, we're able to visualize the data there, and then be able to put it into the decision support tool, which people either would use the website or the phones or any other means of dissemination, because there's no need to look for data. I think data now is everywhere, it's readily available. We are in the era of big data. We need to use this data, satellite data, in situ data, drones, and whatever, to be able to create the systems that are critical for us to, manage our rangelands. Of course, this is really, really an interdisciplinary approach that could make this thing work. We need people working together from ecologists, remote sensing specialists, agriculturalists, all these people. We need to work together to bring the solutions home. There's no one field that can solve all the problem. You saw that the problems are more of the nexus than individualistic. Is climate driven by us? Is biodiversity loss driven by us as people? And then let's contribute to the local, regional, and international initiatives that are focusing on safeguarding our rangelands. I just want to thank a lot of colleagues I work with from the Council for Scientific and Industrial Research, CSR, my former employer, and then also SunParks. I still have solid collaboration there. And then I continue to work closely with some of the scientists to continue with this remote sensing work. I just want to also thank the University of Pretoria and especially the postdocs and postgraduate students that I'm working with. And finally, just want to thank the Grassland Society of Southern Africa for the invite. It's always a pleasure for me to attend this wonderful conference. And then I wish you well. And
Thank you very much for the opportunity to present. My name is Mushe Jomu. I'm a master's student at the University of the Free State, Kwakwa campus. I'll be presenting to you today a chapter from my MSc thesis titled The Impact of Increasing Wood Vegetation on Mountain Biodiversity in the Maluti-Drankensberg region. A case study of 3 species of shrub called Leucocidia cerasea and its nature of encroachment. The project is supported by Dr. Clark, Dr. Torcher, as well as Prof. Leroux. So today, one of the major challenges that our grassland systems are facing globally is the so-called bush encroachment. And bush encroachment is a natural phenomenon where there is an increase, or that describes an increase in woody vegetation in grassland systems. And that phenomenon usually results, or normally results, in biome shift. So this dramatic increase of woody vegetation in grassland systems has been recorded or has led to ecological degradation in a number of cases, as well as rangeland has impacted negatively on rangeland-based livelihood, water provision, fire management, and conservation. So this process, of course, is attributed to several number of drivers, and these include but not limited to the extermination of megafauna. For example, the threat on alien population extinction in the Drakensberg region, or the shortage in big herbivore population in the area that could have probably or possibly led to the extermination of woody species in the area. Looking at overgrazing itself, overgrazing of grasses in grassland systems could mean a reduction of herbaceous layer competition, while overgrazing of woody species could mean more seed dispersal by the grazers in the area, and that is more likely to result in biome shift again, favoring bush encroachment. So reduction in natural field fire frequency, on the other hand, may lead to reduced mortality of woody range expansion or expanding species, as well as reducing grass soil load. That may in turn affect the ecosystem functioning or the response of ecosystem. Of course, we may not rule out that climate change, on the other hand, has also been labeled or flagged as a huge driver of bush encroachment as a result of elevated carbon dioxide. A point of note here is that the previous slides on bush encroachment was to give a brief overview or brief background on how bush encroachment is a problem and what is this bush encroachment we are talking about. But the main subject here is Leucocidia cerasea and its nature of encroachment or distribution. So this Leucocidia cerasea is a key temperate bush encroaching species in the Maluti-Drankensberg region. And this Leucocidia cerasea, again, it's a species that is commonly known as ohot or old wood in English, ohot in Afrikaans, while the Sotho and the Zulu call it chichi and umtiti respectively. As you can see from the picture there, that this is a green species. It is evergreen species indeed. It's a common indigenous shrub to a small tree that is endemic to southern Africa, and it grows up to 9 meters tall and 5 meters wide. So 5 meters wide, it is a big distance for it to cover enough space that could have been occupied by grasses. So this species is species that belongs to the family of rose, and it is a sole species in its genus. Apart from that, it produces yellow flowers with fruits embedded in its old epithylium, which are browsed by cattle and goats. So it is correct to say that the cattle and goats are the seed dispersers of this species. This species is confined to the eastern Great Escarpment from the Snowberg mountain range in the Eastern Cape, south of Inganga in Zimbabwe, as well as Lesotho and Eswatini. And also it is widely distributed in the South African highveld, that is the Free State and the Gauteng province. And in such localities, wide range of its distribution is on damp areas or along the streams, as well as on previously disturbed environment, such as the likes of roads or park or hiking paths, as well as bent areas. Of course, it is not a literature secret that the Maruti Drakensberg, or rather the Drakensberg, is renowned for high floral biodiversity. And I thought the study should at least look at or assess the impact of Lycosidia ceracea encroachment on montane plant diversity in the Maruti-Drakensberg region. And to do that, an objective was set to investigate the impact of Lycosidia ceracea expansion on plant diversity in the Maruti-Drakensberg region. This objective was set on the basis of hypothesizing that Lycosidia ceracea encroachment replaces a rich montane grassland flora with depauperate protoforest flora. So extensive data collection was conducted in the grassland system of Maluti-Drakensberg, and two of those sites were in the Vitishoek Batuba Tribal Authority land, and the other side was Golden Gate Island National Park, while one site in KZN Ukahlamba Drakensberg Park Cathedral Peak Research Catchment number 9. And then last site was at Tsehlanyana National Park in Lesotho. Again, a point of note here is that these grassland systems of the Drakensberg are strategic water source areas for both Lesotho and South Africa. And apart from having high floral biodiversity and endemism, they are famous for ecotourism and commercial livestock production. So any impact in such areas may affect possibly the economy of these two countries. So species richness data for vascular plants was collected at each site. That is the number of species, the number of individuals per species, the percentage coverage per species. That was recorded in Lycosidia thicket and grassland adjacent to the Lycosidia thickets. This was done in a 30 pad 1 by 1 meter quadrant. One set was at Lycosidia thicket, that is the 1 by 1 meter set at Lycosidia thicket. The other 1 by 1 meter was at grassland. And plots in each pairs were separated by 3 meter, and then 10 meter to the next pair of these plots. We of course collected a representative plant specimen in the area or in each study site, and such representative plant specimen was given a tentative name in the field for identification. And of course, all standard barium identification and collecting techniques were followed. For our preliminary data, the analysis, as well as the results, the effect of Leucocydia encroachment on species composition and species diversity of the vegetation in all the sites was considered, with exception of the Lesotho data, since the data is still being cleaned at the moment and is still being processed. Apart from that, the data was analyzed using NMDS, non-metric multidimensional scaling, t-test for Shannon diversity indices. The composition in Cathedral Peak and Bitsisukubatropa tribal area or land was significant with a probability value of less than 0.05. Although there was a weak R-square value of less than 10%, probably affected by the occurrence of Leucocydia, which may be due to old mature Leucocydia, the parent forest that supports an increase in shade tolerant and nutrient species or nutrient demanding species in the area. Our Shannon diversity indices showed no statistical difference in species diversity in both Cathedral Peak and Golden Gate between the Leucocydia and grassland plots. Of course, that was shown by the probability value of more than 0.05. Although looking at the book itself, it showed a significant difference with a probability value of less than 0.05. So it is in this view that the change in elevation between the sites, as well as change in aspects in all the sites, may have probably contributed to these kind of results. In concluding, the results gotten thus far from the study suggest that the encroachment of Leucocydia serratia on montane biodiversity in the Maloti-Drankensberg region does not have an overall impact on plant diversity. However, it is in my view that the type of species occurring in these sites may be impacted by the encroachment of Leucocydia. Hence, going forward with the study, the threat status of the species associated with Leucocydia or open grassland may still be something that one may consider, and simply because I believe that may provide an additional insight into the conservation implication of the wood encroachment in these grassland systems of the Maloti-Drankensberg. Thank you so much for the opportunity once again.
Hi everyone, my name is Yonela Maziko from the University of KwaZulu-Natal. Today I'll be talking to you about the influence of drought and fire on grass sward composition and structure in a South African mesic grassland. The total Earth's surface is covered for up to 37% of grasslands. In South Africa, a large percentage of that, which is about 60%, has undergone deteriorative transformation in relation to climate change. Fire can also be attributed to the terrestrial changes that we see today, as it is a vital regulator of ecological diversity. Bringing us to our main question: how does fire interact with the changing environment to bring about the conditions that we see today? The objectives of the study are to determine the influence of drought on species composition under different burning regimes. It is also to quantify the effects of interactions between fire and drought on plant community properties. Our research questions. What effect does a short-term drought have on species composition under different burning regimes? And does an interaction of fire and drought between treatment spark a response of plant communities under drought conditions? Dry conditions are projected to have a significant impact on the composition and structure of grassland ecosystems. So with the limited information that is currently there now on how drought affects various species dynamics in conjunction with fire, it is imperative to attempt to understand the extent and pace of changes in species composition and function through field-based trials like this one. It is also important to establish the impact of the combination of fire and drought on plant communities, as the uncertainty of rainfall reliability warrants further investigation on the influence of drought and fire on ecosystem dynamics such as species composition in order to provide essential understanding that will further improve the overall SWOT health of grasslands. Bringing us to our methodology. The study site is located at Ukulina Research Farm, which is in Pietermaritzburg in the province of KwaZulu-Natal, South Africa. The area generally has an average rainfall of around 690 millimeter per year, with warm summers that average monthly maximum temperatures of about 26.4 degrees Celsius and winters with an average minimum of 8.8 degrees Celsius per month. The veld is located as a grassland biome. The study is based on long-term trials that were established in the 1950s, with burning treatments at different frequencies and seasons of application. 3 different treatments are burning, the annual burn, and the triennial burn, which occurs in spring, and they are all replicated 3 times in a blocked design to account for any soil depth changes. This is how currently the treatments or the site looks. The treatments were surveyed from 2005 to 2010 for species composition, after which an impactful drop in rainfall was noticed, and then the treatments were resurveyed again during the early and late growing season of 2019 or 2020. They were resurveyed for species composition to compare any changes that were brought about by the short-term drought that could have taken place since the previous vegetation survey. Comparable experimental treatments and sampling procedures were used in assessing the effect of fire and drought on composition, richness, and diversity of plant communities, whereby 4 meter squared quadrats were divided into 4 quadrats, and they were relocated at the same positions as the previous resurveys for species composition assessment. This is a pictorial representation of the rainfall patterns around Ukulinga Research Farm over a period of about 20 years. As you can notice that the rainfall started to gradually decrease from the year 2010, falling well below the average or normal annual rainfall, which was stated earlier in the area, especially around the years 2015 to 16, and again in 2019, which leads us to believe that there is a drought occurrence around this time. Statistical analysis. Differences in species composition were assessed using the canonical correspondence analysis, and then variations in species richness, biodiversity, together with evenness were analyzed with a two-way ANOVA, and further analysis was done to determine the variations between treatments using a Turkish test. Our results showed us that the annually burned treatments had a higher concentration of grass species when compared to the other treatments, with the unburned areas mostly dominated by wood species instead of grass species and forbs. In terms of species diversity, the results found that there is a significant main effect of year on species diversity. It is significantly higher in the years followed by the drought. It was also found that the treatment has a significant effect on species diversity when annual burning was higher in terms of diversity compared to the unburnt and tri-annual burnt treatments. When it comes to species richness, the year significantly affected species richness where it is higher in the year that follows drought. Unlike the burning treatments also significantly affected species richness, whereby annual burning also had a very high species richness compared to the control and also the triennial burning. With regard to species evenness, unlike in diversity and richness, drought showed no significant effects on species evenness between the years, but there was a significant impact on species evenness between burning treatments. It is evident that burning treatments were significantly affected by drought, whereby there was no interactive effect between burning treatments and drought. Concurrently with the previous research papers published on this site, we can safely conclude that frequent burning significantly favors a higher diversity and also richness of species. I thank you.
Good day, ladies and gentlemen. My name is Manjana Matule. I am an MSc student from Northwest University under the supervision of Prof. Chris Munyati. Dr. Aileen Yudan was responsible for some analysis, and she's also from Northwest University. My presentation is titled The Effect of Proximity to Highways on Above Ground Tissue Surface pH of Gray Savanna Grass Species. This is the outline of my presentation. It begins with the introduction and outlines the context and scope, methods which would also outline the study area, fraud sampling, and the analysis of the samples. Result will give information on what was obtained in the experiment, and it will be followed by discussion and conclusion, and lastly, acknowledgements. The palatable grasses in the savanna rangelands grow in close proximity to road. The pictures depicts the grasses growing in close proximity, both in the Zeerust Road and the Satara Road that is in Kruger National Park. In those roads, you have motor vehicles that emit gaseous pollutants such as sulfur dioxide, carbon monoxide, and oxides of nitrogen, and heavy metals such as your lead, cadmiums, and etc. So according to a study by Ashenden and other authors, these pollutants can have a negative effect on plant growth and susceptibility to stress. So there's a little understanding on how these pollutants affect the savanna grass species. In the Karoo, a study by O'Farrell and Moulton showed that roads can affect grass species diversity. The aim of this presentation is to assess the effects of proximity to highways on tissue surface pH in palatable savanna grass species and the possible effects on grazers. This study was conducted in South Africa, preferably in Gauteng province in the city of Pretoria and Northwest province in the city of Mahikeng. In Pretoria, the study was conducted in the provincial road R80 at N4 junction, and in Mahikeng it was conducted in the national road N18 in two transect T1 and T2. Transit 1 is closer to Mahikeng Central Business District and other roads. Transit 2 is an open area and is closer to the Crossing Mall in Mahikeng. The control site was in native woodland, Northwest University at Mahikeng campus. The R80 road was a high traffic treatment site, and the grass patches along the road is where the grass species were collected. The N80 road was a low traffic treatment site, and the grass patches in the below picture is where the grass samples were collected. The control site was the native woodland grass, Northwest University, and it was away from the roads, and the grass species samples were collected at Mahikeng campus. There were three palatable grass species that were studied. The first one is Digitaria eriantha, commonly known as finger grass, giant or Pongola finger grass. It is considered as a very high nutrition value species with very high amounts of your nitrogen, phosphorus, potassium, and magnesium. It has large leaves, 2 to 7 millimeters wide, 5 to 20 centimeters long. It has digit-like flowers, and it is also considered as a climax decreaser species, and it is most preferable as species for livestock because of its sweetness. The second species is Brachyra nigopredata, commonly known as the black-footed grass. It is a selectively moderately high nutrition value species with high amounts of nitrogen, phosphorus, potassium, and magnesium. It has large leaves, 2 to 8 millimeters wide, 6 to 30 centimeters long. It is also a climax decreaser species, and it is considered a good grazing value species for livestock. And the last species is Erogrestus lemoniana, commonly known as the lemon lovegrass. It is selectively low nutrition value species with low amounts of nitrogen, phosphorus, potassium, and magnesium. It has small narrow leaves of approximately 1 to 3 millimeters wide and 2 to 40 millimeters long. It is a subclimax increaser species. It is native to South Africa and the southern region part of Africa, especially the SADC region. It is considered an invader species in some continents like America and countries like United States of America. Field sampling was conducted on 1 to 3 March 2021, the summer season, when grasses were at their full maturity. The highway was perpendicular to the transects. The sampling distance from the road edge was 0 meters, 20 to 30 meters, 50 meters, 60 meters, and 100 meters. The transect at R80 was less than 50 meters, and it was limited by fences, and up to 100 meters at N18. Digitary Ariantha was absent at N18 road. A minimum of 3 to 5 whole stem grass tissue sample was cut from the base from 3 random plants per species as depicted by the picture above. Same procedure was done in the control site from 3 sample sites per species. Samples were transported the same day to the laboratory. In the laboratory, leaf length were measured at an average of 3. For each sample, grass tissue samples were randomly selected in a plastic bag and placed in a new plastic bag and added 40 milliliters of distilled water, shook for 30 seconds, then transferred the washed off materials in a glass beaker. Before measured waters, the pH was calibrated using the pH meter, and that is depicted in the picture above, and also the buffers, and verified to be 0, and that's when the pH was measured. So the same procedure was done for electrical conductivity, but with different grass tissue samples using the electrical conductivity meter below. Leaves were cut and stored in a honey jar fully submerged in 70% ethanol and transported in a specialized laboratory for scanning electron microscope for stomata trachome counts in 100 microns blocks. And just for a reminder, stomata is responsible to regulate the flow of gases in and out of leaves and thus the plants as a whole. Trachome is responsible for physical and chemical protection for the leaf against microbial organisms and insects. What was observed in the results was that grass tissues close to the highways have low pH and higher pH away from the road veg. This was observed in both Brachyara negopredata and Erogrestus lemoniana, wherein the trend lines are increasing as the distance increase. And if the distance is increased in both graphs, let's say to a distance of 200 meters, it will equal to the control site averages, but it was less pronounced in Digitare Ariantha. The statistical significance of difference between the pH and the control site is that at a distance of 0 meters, the 3 species displayed statistical significance with varying percentage, with B. nigopredata at 0.1% and both D. ariantha and E. lemoniana at 5%, and a distance of 20 to 50 meters, the statistical significance was observed in B. nigopredata at 0.1% and E. lemoniana at 1%, with the exception of D. ariantha, that was above 5%. Grass tissues close to the highways have high conductivity and lower conductivity away from the road veg. This was observed in both Digitaria eriantha and Erogrestus lemaniana, wherein the trend lines are decreasing with an increase in distance, and if the distance was increased, the trend lines was going to be equal to the control site averages, and it was less pronounced in Brachyura negopredata. The statistical significance of difference between the pH and the control site is that at a distance of 0 meters, both Digitaria eriantha and E. lemaniana are at 5%, with the exception of B. negopredata above 5%. At a distance of 20 to 50 meters, it was only E. lemaniana at 0.1%, whilst both B. negopredata and E. lemaniana above the significant percentages. Just to show how the stomata and trichomes looks like for Brachyura negopredata in the 100 micron block under 1500 times magnification. The trichome was observed only in the control site macrograph, and the stomata was observed in both the control site and the road veg macrographs. The epidermal cells on the road veg sample had dust particulates or pollutants, which was not observed in the control site sample. The stomata for Erogrestus lemaniana in the 200 microns block under 800 times magnification was observed in both sample macrographs. The epidermal cells on the road veg sample had dust particulates or pollutants, which was not observed in the control site sample. Micromorphology attributes in 100 microns area. It was observed that the number of stomata in control site is higher than in other locations, with the exception of a location at a distance of 30 meters for Brachyura negopredata. So the number of trichomes in control site is higher than in other locations, with the exception of location at a distance of 60 meters, which shows an anomaly. So the stomatal frequency and index at a control is higher than in other locations. What caused the grass tissue samples close to the roadsides to have low pH was due to sulfuric acid and nitric acid that was emitted by vehicles. The acidity on leaves might have been reduced by dilution by rainfall before sampling dates, carbonates in dust, which tend to have high pH. A 4-month study was conducted in Switzerland, and it showed acid pH on roadside tree leaves. Therefore, more sampling is required to be done over a period of time, not just once in the study area. High leaf electrical conductivity was caused by metallic ions such as lead, cadmium on leaf surfaces. Low pH can have the following negative effects on grazed grasses. Firstly, reduced growth due to morphological disturbances, slight change in the taste of the grasses since they differ in their nutritional content values. Acidity can reduce mineral metabolism by grazers. The effects seem to vary by grass species depending on leaf characteristics. What are the environmental management measures required to lessen the effects? Firstly, low road density in grazing areas. Even though this might have negative effects on tourism in places like Kruger National Park, that is depicted in the picture, and also agriculture. Secondly, cleaner motor vehicles. So vehicles with low emission levels or rate will be most preferable, and electrical cars, since there wouldn't be the emission of pollutants. And thirdly, cleaner fuel, so which is unleaded petrol will be most preferable than leaded petrol. And lastly, you have enforcement of legislation on vehicle emission standards. In conclusion, proximity to roads has a negative effect on the quality of palatable grasses in savanna rangelands. Most palatable species of savanna grasses are vulnerable to the negative effects of proximity to roads. These are the references of my presentation, and some of these studies were done in eastern regions of the Free State and in Mpumalanga in South Africa, and it was published in the South African Journal of Animal Science. I would like to acknowledge the Northwest University, the Department of Forestry, Fisheries and the Environment for sponsoring my studies, and I would like to extend my sincere gratitude to everyone who took an opportunity to listen to my presentation. And once more, thank you for.
Good day, everyone. Title of my talk is the value of remotely sensed vegetation indices and animal movement patterns for rangeland management in the Upper Karoo. Remote sensing techniques offers a non-destructive and a more cost-effective alternative that can produce more timely and more precise data. There is a need for a quick, reliable assessment method in order to determine a species composition, your vegetation coverage, and what is a potential driver of livestock movement patterns. Therefore, the aim of our study was to better understand the spatial relationship between vegetation composition, vegetation activity, and the movement patterns of sheep within different grazing camps. Our study area is located within the Northern Cape Province of South Africa. Here you can see the boundaries of the Northern Cape Province. In green is where the Nama Karoo biome is located within the province, and in orange is where our study area is located, called the Western Upper Karoo vegetation type. The stocking density trial was started in the year 1988 by the Department of Agriculture, and it consists of 4 stocking densities. It's a high stocking density at 4 hectares per small stock unit, recommended at 5.5 hectares, light 7 hectares, and a very light at 8 hectares. This picture was taken on the boundary at the stocking density trial. Here's the fence line. Everything you see in front of you is where the stocking density trial is located. Back in 1988, they selected this specific site because they thought it was the most homogeneous area in terms of vegetation composition, and they did not have access to satellite imagery back in 1988. But there is actually drainage lines running straight through the stocking density trial, and there where that yellow line is located is actually a drainage line running straight through the trial. The picture I just showed you was taken right here on the boundary of the stocking density trial, looking in this direction, and the yellow line I showed you was this drainage line running through the 3 camps. There's a second drainage line running through these camps here in the back, and we monitor it with satellite imagery to determine what's the vegetation activity. Green would indicate very high vegetation activity, red would indicate very low vegetation activity. We could see that the area was actually not that homogeneous as thought back in 1988 when they selected this specific area, because we could see that there were drainage lines running straight through the different camps. We monitored it over time. Moving on to end of November, 4th of December, we could see they received no rain during this time, leading up to the rainy season, and vegetation conditions only deteriorated. It was at its worst here on 13 January 2018. You can see the red indicating very low vegetation activity, and then at the end of January they received some rain. What was interesting then is to see that these areas in the drainage line, the vegetation reacted much quicker compared to the areas right outside of the drainage lines to this rainfall that they received. 5 or 10 days later, you can actually see it's starting to green up now. But the areas that reacted first to this rainfall was the areas in the drainage lines, and the areas outside of the drainage lines still indicated very low vegetation activity. To determine what was the cause of this, that certain areas in the camps reacted quicker to the rainfall, we did soil depth analysis on a 100-meter grid across the entire study site. And we found that in these drainage lines, which reacted quicker to the rainfall, the soils were deeper at 40 to 50 centimeters deeper, significantly more compared to the areas just outside these drainage lines, where it was only 5 to 15 centimeters deep. I wanted to track it over a 3-year period to see if there was really a significant difference between a deep soil and a shallower soils. So here you can see the deep soil is the green line, we have the shallow soil with the brown line, and in blue bars is the rainfall. So this was for a 3-month period, the rainfall that they received during that period. And you can see it was significantly higher compared to the shallower soils. And even a few weeks later, when the vegetation started to react to this good rainfall, we can see that it outperformed the shallower soils in terms of vegetation condition scores. Especially we see it at the start of every rainy season from January up to May. We can see when they receive rainfall that there's a big spike in your NDVI values in the deeper soils compared to the shallower soils. So to find out if the sheep know that there's certain areas in the camps that react quicker to the rainfall compared to other areas, I had to make use of GPS collars. It was set on a 3-hour interval where the GPS position was captured. There was 3 GPS collars per herd. That's a total of 12 GPS collars. And to date, we have more than 3+ years of GPS collar data. I also work with the cloud-free Sentinel-2 imagery running from 2016 up to 2020. I also worked with a total of 8 vegetation and moisture indices. There you can see the indices listed, and this was to also find out what is the best index in this vegetation type, the Karoo shrublands, to work with to identify these areas that react quicker to the rainfall compared to other areas. Okay, so to start, we have more than 100,000 GPS points recorded over this time period, so I needed to clean up. What I did is remove the areas around the watering points. I needed to remove these GPS points because the animals spend quite a bit of time during the day around the watering points, and so I don't want to identify that these are the areas that they spend most of the time with. So we clipped these areas, and we also clipped the data at the boundaries where the sheep tend to go and lay down. This is typically what we saw during high windy days when the wind direction would typically be from the north. The animals would move in this direction, hit the fence, and they would just go and lay down there and spend the day there. So we also didn't want to identify these areas as their grazing sites. So to determine if the animals do spend more time in certain parts of the camps, I work with optimized hotspot analysis in ArcGIS. The GI statistic was to identify statistically significant hotspots of preferred grazing areas. So the input field would be the GPS data, use the count incidents within a fishnet polygon, and then provided a bounding polygon to identify or define the count boundaries. And then the GI tool and analysis each feature and determines whether the feature is a hotspot or not. So we get a Z-score to determine the statistical significance, whether it's significant or not significant. So to visually show you how the data would look like and the analysis of this data is here you can see if we focus on this camp A2, this is the NDVI in the background. So here you can see higher vegetation conditions in the drainage lines, and this camp actually had no drainage line running through it. And so here you can see where all the GPS points was recorded. And if we run then the hotspot analysis, you can see across the entire camp it's only gray blocks. So gray blocks indicate that there's no significant areas where the animals spend significantly more or significantly less time. So they're utilizing the entire camp. If we move over to camp B2 now, you can see that there's a small part of this camp where the drainage line is running through with higher vegetation condition points. Remember that the watering point was here where they spent most of their time. There we have the GPS points. If we run the hotspot analysis, we can see now in bright red here showing with 99% confidence they're spending significantly more time in this small corner of the camp. So where they have available grazing the entire camp, they're spending most of their time in this small corner. Moving next door to C2, now we can see the drainage line running straight through this camp with high vegetation conditions. Again, the GPS points. Running the hotspots analysis, it shows that they spend significantly more time in this area where the drainage line runs straight through the camp. Where it shows up in blue, you can say with 99% confidence they're spending significantly less time in these areas where the drainage line is not located. Another good example is Camp C4. This is the biggest camp that they in the stocking density trial under the light stocking density. So these animals have a larger area available to graze. And we can again see just this small corner here in the right is where the drainage line runs through this. We have the GPS points, run the hotspots analysis, again spending significantly more time in this area here. But this is important, especially for farm managers and farmers, is you may have the idea that you have, say, a total of 200 hectares available, a camp of 200 hectares, but the animals are only utilizing 28% of that 200 hectares. So they're overutilizing and trampling this area here and not utilizing the rest of the camp. Okay, then doing the index comparison, as I said in the beginning, as we assessed 8 different moisture and vegetation indices, you can see there were a few good correlations between the green NDVI, the green coverage index. There were good correlations, but the best one was found to be the NDVI. The highest correlation between the index value and the Z-score from the hotspots analysis was found to be NDVI. Okay, and as mentioned in the beginning, we also wanted to identify the best spatial resolution with the strongest correlation for NDVI and the grazing hotspots by using a pixel comparison analysis. We worked with a 10 by 10 meter pixel. This is what you typically would get from Sentinel, as I said, every 5 to 10 days. 20 by 20, 30 by 30, up to 1000 by 1000 meter pixel sizes. Here you can see the 1 by 1 kilometer pixel size is quite big, and you aren't able to identify where these drainage lines are running through anymore. We found that the strongest correlation was at 100 by 100 meter pixel size, and the drainage lines with the higher NDVI scores were still clearly visible here at 100 by 100 meter pixels, but they become less detectable if you go larger, up to 250 or 500 by 500 meter pixels. This is just to show you the results from the analysis that the best correlation was at 10,000 square meter. Okay, so to conclude, the hotspot analysis revealed statistically significant clusters of sheep in areas with higher vegetation conditions. These preferred grazing sites did vary between seasons and between camps, and we saw that camps without a water drainage line in the deeper soils, no grazing preferences, and the sheep tend to utilize the entire camp. Where there is a drainage line running through the camp, it results in a significant hotspot in and around these areas. And the farmers need to be cautioned, as this may result in overutilization of these areas. From the 8 indices that we analyzed, we found that the NDVI was the most effective index to indicate the clustering of these sheep. The spatial resolution was found that the strongest correlation for NDVI and grazing hotspots were established by using a pixel comparison analysis. We compared the different pixel sizes, and the strongest correlation was found to be at 10,000 square meter pixel size. And we saw that drainage lines with a higher NDVI score was clearly visible at higher spatial resolution, but they become less detectable when your resolutions are above 62,500 square meters. So historically, understanding of and measuring livestock movement patterns in the semi-arid rangelands of the Upper Karoo was a time-consuming and a very costly process, but this study has demonstrated the effectiveness of remotely sensed vegetation condition data that can be used as a grazing management tool. By understanding the spatial variation of plants within camps, it can be used in planning of farm infrastructure, such as fencing and water reticulation, in order to better manage and prevent overutilization of these areas. So a way forward to this study would be to look at the animals' weight data. They are weighed every 2 weeks, and it's been done for the last 30 years, and determine if there is a correlation between animal weight gain and vegetation conditions that we get from satellite imagery. We want to also look at animal movement patterns and determine if the daily distance walked by the sheep in the different stocking density do differ or not. Thank you very much.
Hello everyone, my name is Musilanjara Shao from National University of Lesotho. It is my pleasure to take you through my presentation, which is on perceptions of rangeland users on the ecology, socio-economic impacts, and management of invasive plant species, a case of Mpaki in Gauteng district, Lesotho. Why should we be concerned about rangelands? Rangelands are land ecosystems that are characterized by herbaceous and shrub cover. These ecosystems support livestock production and the supply of ecosystem services such as water. However, these ecosystems are threatened due to unsustainable use of resources and soil erosion, among other factors. It is important to note that human beings are part of the rangeland ecosystems, and they are also causing this deterioration. It was therefore important to understand their perceptions. The objectives of the study were to evaluate possible introduction pathways and methods of spread, to assess the impact of invasion on rangeland condition, to determine socio-economic impacts, and to investigate management interventions. The study was conducted in the rural areas of Gauteng, which is one of the 10 districts in Lesotho. Questionnaire and focus group discussions were used for data collection. 190 respondents were interviewed. These informants were asked for consent before administering the questionnaire. The focus group discussions involved farmers' associations. The variation in the rangeland users comes from an understanding that different rangeland users have different relationship with the rangeland depending on the benefits derived from the rangeland. SPSS was used to analyze the data. About 8 questionnaires were discarded because they had a lot of data gaps. I now present findings of the study. This figure shows the common invasive plants occurring in the area. Artemisia afra and Leucocidia sericea were surprisingly listed among invaders. These plants are not documented as invasive in Lesotho. However, the assumption is that local communities perceive any plant as invasive once it becomes problematic. Under Objective 1, it was found that there are 2 sets of invasive plants, the native and the alien. The respondents indicated that the latter comes from urban areas and they are spread by human activities, natural factors, and livestock during browsing. This can be seen on the picture where a goat is browsing on fruits of Rosa rubiginosa. This goat is going to spread the plant through internal and external means. This figure shows different agents of spread for various invasive plants. It can be observed that human activities is the most dominant method of spread across all invasive plants. Under Objective 2, the respondents had a consensus that indeed rangeland condition has deteriorated. It can be seen that plant invasion, climate change, and overgrazing are the most implicated causes to the deterioration of rangelands. Under Objective 3, about 7 aspects are impacted negatively by plant invasion. This figure shows that Rosa rubiginosa is dominant across all aspects, with over 55% on most of them. This picture shows invasion by Rosa rubiginosa on the rangeland, and total rangeland productivity is reduced. On the left is a goat with entangled mohair, and this compromises the quality and quantity of mohair. Another typical example is on the left picture, we see contaminated wool on the floor, and it was infested by Xanthium strumarium. And the farmers have to remove this waste before exporting the wool and mohair. And this is said to affect the grades of this product. This figure shows that sheep are the most reared livestock and the most affected type by plant invasion. Although Rosa rubiginosa has negative ecological impacts, it also has positive impacts to the communities. This is evident on the picture where a grandmother had just arrived at the spot to harvest the fruits. The respondents indicated that a 20-liter container full of these fruits is sold for 100 maluti, which is equivalent to about 7 USD. These fruits are sold to commercial firms for processing into other value-added products. The last objective, it was found that the method of control is mainly physical removal of invasive plants. It was found that different stakeholders are participating in the management of invasive plants, and mainly these are the community and the government. However, it is worth noting that over half of the respondents indicated that they were not aware of any management interventions in the area. The respondents suggested different methods of control, including employing the communities and herders. The herders were described to have human power, free time during herding, and they are the least developed group in the societies. Therefore, they could be hired to remove invasive plants. During the suggestions, two main responses were observed, where the rangeland users without livestock were suggesting partial removal of invasive plants, whereas those who have livestock were suggesting eradication of invasive plants. In conclusion, the study has found that Rosa rubiginosa is the predominant invasive plant with most negative impacts. Major agents of spread are human-induced activities, wind and water erosion. Rangeland deterioration in the study is mainly caused by overgrazing, plant invasion, and climate change. All aspects of deterioration contribute to the ultimate reduction in economic value. Management in the area is mainly physical removal. There is a conflict of interest on removal of invasive plants. Recommendations. For effective management of invasive plants, the study recommends that people at grassroots level should be involved. Policymakers should consider the interface between social and ecological systems. Lastly, more studies on invasion ecology and social impacts of invasive plants are recommended. I wrap this presentation by acknowledging different participants in the study. I thank you for your time. Please stay.
Good day, everyone. My name is Pete Munehi, and I'll be giving a talk on the density effects of mature trees on grass production and tree seedling recruitment in a bush and coast savanna. Firstly, I would like to acknowledge my co-authors. Many rangelands are affected by the problem of woody plant encroachment. Woody plant encroachment is the proliferation of trees at the expense of the herbaceous layer, which may negatively affect livestock and game farming. Given the negative effects of woody plant encroachment on pastoral productivity, ecologists and land users have often considered tree thinning as a control measure. Tree thinning may result in increased canopy gaps that may release the grass layer from the suppressive effects of woody plants. Increased or enhanced grass production may also lead to the reduction of seedling recruitment. Increased canopy gaps may also stimulate the growth of the remaining mature trees, which may enhance soil resources. However, tree thinning success may depend on numerous factors. Studies have demonstrated that the effects of tree thinning differ even within similar environmental conditions. Site-specific drivers such as plant species and soil types may be responsible for these variations. Few studies have compared the response of rangelands to tree thinning among different woody species, which leaves the question of whether differences in plant traits influences the ecological or management outcomes of tree removal or removal methods. For instance, multi-specific stands usually have a higher productivity compared to monospecific stands. Mixed tree species improves resource usage compared to pure stands by improving the supply and capture of resources. In addition, plant traits may be an important determinant of tree thinning success. For instance, woody species such as Vachelia totilis enhance water infiltration and soil nutrients inside their canopies rather than outside the canopies. Thus, this suggests that the canopy gaps created through tree thinning may not be beneficial in increasing the overall herbaceous biomass production in a Vachelia totilis monospecific stand. In addition, soil texture may also affect plant growth. Soil texture alters water intake rate, storage, and aeration. Studies have shown that sand-dominated soils allow for rapid infiltration and permeability of soil water. In clay-dominated soils, infiltration may be affected, which may increase runoff or water logging. Also, because water is more difficult for plant to extract from clay soils than from sandy soils, when soil moisture content is low, soil texture could alter tree and grass physiological responses to soil moisture variability, aggravating water stress when soil moisture fluctuates. Thus, differences in plant species and soil texture could be an important determinant of ecological services after tree thinning. In this study, we evaluated the effects of different tree thinning intensities on grass biomass production, tree seedling establishment and growth, and the growth of the remaining mature trees. We predicted that grass biomass production will increase with increasing tree thinning intensities because of the reduced competitive effects from woody plants. We also predicted that Tree thinning will reduce the establishment of tree seedlings because of an increase in grass biomass after tree thinning. Lastly, we predicted that reduced tree competition will significantly increase the growth of the remaining mature trees and also tree seedlings. The current study was conducted at the Agricultural Research Council at Roodeplaat Farm that is situated in Pretoria. In this study area, we used two study sites. Study site 1 was in clay-dominated soils encroached by monospecific stand of Vachelia totilis plants, and study site 2 was in sand-dominated soils encroached by multiple woody species. At each site, we set up 24 plots of 30 by 30 meters that were subjected to different intensities of tree thinning. In study site 1, we found that grass biomass production was negatively affected by 75% removal of trees and 100% removal of trees during the first growing season, whereas there was no significant changes during the second growing season. In Site 1, we also found that seedling establishment and growth were not significantly affected by tree thinning. However, tree thinning significantly increased tree growth, particularly in 50% and 75% thinning treatments. In Site 2, grass biomass production increased after the first growing season, with significant increases observed in 100% thinning treatment only. Also, after the second growing season, grass biomass increased across all treatments, with significant increases observed in 50%, 75%, and 100% thinning treatments. Inside 2, seedling establishment was significantly reduced in 50%, 75%, and 100% thinning treatments, although we found no significant changes in seedling growth. In addition, tree growth significantly increased in Site 2, although the changes were observed on tree height only, with trees that were in 50% and 75% increasing in tree height. We attributed the diminished grass biomass production in Site 1 to that Vachelia totilis trees do not facilitate the herbaceous biomass outside their tree canopies. Also, we observed that during the study period, high intensities of tree thinning led to the increase in soil erosion. Soil erosion is known to reduce the herbaceous cover and the soil seed bank that is necessary for vegetation redevelopment. Therefore, our results suggest that plant characteristics and soil texture may be an important determinant of tree thinning success. For example, Smith reported a recovery of the herbaceous layer after tree removal in eroded sand-dominated soils, a result consistent with our findings from Site 2. Nonetheless, our results were consistent with Bailey, who reported that the removal of trees results in a reduction of the herbaceous biomass, and also Allegretti, who reported negligible results after tree thinning. Nonetheless, our results from Site 2 are consistent with the notion that tree thinning results in increased grass biomass. We attributed the lack of significant changes in seedling establishment in Site 1 to the undetermined factors such as the predation of Vachelia totilis seeds by insects and a possible reduction in the soil seed bank, possibly caused by rainfall-induced soil erosion in the study site. Nonetheless, the results from Site 1 were consistent with the results of Brodvik and Asbjørnsen, who showed that tree thinning did not have any significant effects on the density of white oak seedlings. We, however, attributed the results from Site 2 to the higher grass biomass production after tree thinning in 50%, 75%, and 100% thinning treatment, which may have possibly suppressed the establishment of tree seedlings. The lack of significant difference in growth response of the tree seedlings could be attributed to the negative and positive responses of the herbaceous layer after tree thinning in both study sites. In Site 1, for example, diminished grass production across all treatments may have provided seedlings with an equal opportunity for growth, resulting in the lack of substantial differences in seedling growth among the treatments. In Site 2, the increase in grass biomass across all the treatments may have suppressed the growth of seedlings in all treatments. Our data from both study sites were consistent with the prediction that trees in 50% and 75% in treatments will significantly increase in growth. These results can be attributed to the reduced competition in 50% and 75% thinning treatments. Also, these results are consistent with the results from other similar studies that show that the removal of neighboring trees results in greater growth of the remaining trees. In conclusion, tree thinning had contradicting effects on the grass layer in this study. However, the results from Site 2 suggest that an optimal tree density to maintain in savannas can be achieved through tree thinning. We suggest that future research with the inclusion of grazing animals will be vital to identify the long-term management options for controlling woodplant encroachment while promoting the herbaceous layer. Also, increased or diminished grass biomass played a vital role on seedling recruitment in this study. In addition, reduced tree competition significantly affected the growth of the remaining trees. Therefore, the removal of some of the woody material may release the remaining individuals from competition-induced size limits. The overall implication of the study findings is that regardless of the substantial cost of woody plant control, the recovery of key ecosystem services such as an increase in forage production may be absent. However, this may be system-specific and therefore in other systems, the absence of management interventions such as tree thinning may compromise ecosystem services and function. Thank you very much for listening.
Good day, my name is Clement Kipido and I'm presenting on behalf of one of our students, Tariq Jamali. Tariq is a current master's student at the University of the Western Cape. And I will be presenting findings from one of his chapters in his thesis titled The Diversity of Poisonous Plant Patches in the Arid Rangelands of Namaqualand. Poisonous plants are distributed worldwide and are found in almost all plant communities. They pose a significant economic risk to the livestock farming industry, as livestock losses may occur as a result of ingestion of these plants. Ingestion may result in livestock fatalities, decreased productivity, and increased management cost for farmers. As a preventative measure, some farmers may want to remove poisonous plants from the landscape because of the risk they pose to livestock. However, the full extent of the ecological consequences of removing these plants from an area remains unknown. Thus, understanding the diversity of poisonous plant patches in relation to the rest of the landscape will ultimately lead to a better understanding of the role that poisonous plants play in these ecosystems. Where there is limited research on the role of poisonous plants in driving the diversity of the landscape, research does exist on how certain plants facilitate the growth of others, owing to their size and age, which may in turn drive diversity. These plants are called nurse plants, as illustrated in the picture, as they shelter plants that are called beneficiaries from biotic and abiotic stresses. Nurse plants are commonly found in environments in which herbivory or abiotic stresses limit plant growth and the ability to reproduce. This type of positive species interactions play a major part in increasing species diversity and structuring plant communities. Nurse plants may directly or indirectly influence beneficiaries in a variety of ways, creating the optimal conditions for facilitating by ameliorating conditions beneath its canopy, allowing for better seed recruitment, germination, and growth. But few studies have addressed how herbivory may affect plant interactions. Research has shown that in environments in which water is limited, the effects of grazing on plant performances can be very important. Owing to lower vegetation productivity in water-limited areas, the available palatable species may experience higher grazing pressure, in which case the dynamic of nurse plants providing protection against herbivory may be an integral part of maintaining diversity in water-limited environments like the arid rangelands in Namaqualand. Thus, this study considers the effect of poisonous plant interactions on diversity, arguing that poisonous plants act as nurse plants by deterring herbivores. There has been little to no research done on the influence of poisonous plants on diversity. This study tested the hypothesis that poisonous plants not only protect palatable plants from grazing, but act as islands of diversity in an arid grazing system. Specifically, the study aimed to assess the association between poisonous plants and palatable food species, and to determine whether poisonous plant patches could be considered islands of diversity. The study was conducted in the Steinkopf communal area, which is just less than 600,000 hectares in size, and there's about 50,000 resident pastoral livestock that are herded from permanent to temporary stock posts, which are scattered throughout the rangeland. Within the Steinkopf communal area, 25 sites were selected in the winter rainfall shrubland and were based on patches dominated by large poisonous plants within the genus Tylocodon and Euphorbia. These patches were found along grazing routes recommended by herders and livestock farmers in the area. At each site, which was 225 square meter plots, it was laid out, which was further divided into 20 subplots using 2 by 2 meter quadrants. Within each quadrant, plant species were recorded, specifically the name and number of occurrences. Each recorded species was then placed into functional groups based on palatability and growth forms. This process was then repeated for non-poisonous plant patches that would mean in the matrix in between these sites, and each of them were 25 sites. Diversity was calculated using the Shanawinan index. Statistical analysis on this data included independent sample t-test comparing the diversity of species sampled on poisonous plant patches to that of species sampled in the matrix. Similarly, a t-test comparing the diversity of functional group on and off poisonous plant patches was also conducted. Furthermore, Cole index was used to determine which species are associated with poisonous plants found on poisonous plant patches. This association is based on the degree to which these species' joint occurrence become more frequent. The results in this figure indicate that poisonous plant patches have significantly higher mean alpha diversity than the surrounding matrix. In this case, the mean matrix species diversity was 1.34 and the mean poisonous plant patch diversity was 1.94, with the t-test being minus 5.454, and the result was highly significant. From the result, it became apparent that poisonous plant patches have significantly greater mean alpha diversity than patches that occur in the matrix. Furthermore, poisonous plant patches have a significantly higher mean growth form diversity than its surrounding matrix. And in this case, the mean matrix growth form diversity was 0.94, and the mean poisonous plant patches growth form diversity at 1.46. And the t-test result was minus 7.34, and that was also highly significant. From these results, we may consider poisonous plant patches as islands of diversity. Diversity may be driven by patch dynamics, where resources are concentrated on patches as a result of redistribution of water, seed, and nutrients in the landscape. The conditions in these patches may facilitate growth, allowing for the establishment of vegetation. The diversity in these patches may also be driven and maintained by the presence of poisonous plants. These plants may act as a deterrent for livestock or herbivores in general that may potentially graze on the palatable plant species on these patches, thus preferring rather to graze within the matrix on certain species. By preventing herbivory, poisonous plants may allow for the establishment of a greater diversity of species and growth forms on patches. The 5 targeted plant species are poisonous to livestock, which I will illustrate in the next slide, and are typically avoided. A Coles index indicates that plants associated with these 5 targeted species across the 25 sites sampled: Adromyces, Euphorbia, Tylicodon paniculatus, and Tylicodon reticulatus are highly associated with species in the genus Lampranthus, a small or dwarf succulent, with the exception of Tylicodon wallichii, which is highly associated with species in the genus Pensea. The positive association observed using the Coles index may be attributed to the aforementioned nursing effects, in which the target poisonous plant species having a positive effect on the associated species. The target species may have modified the patches in such a way so as to facilitate the growth of the associated species, and this was work done by Suraija 2005. Alternatively, these associations may be due to an overlap in environmental tolerance variables, ultimately forcing both species to occupy the same area. An understanding of which palatable species are associated with poisonous plants may allow herders to better identify areas where poisonous plant species occur, as some palatable plants stood out more from a distance than some poisonous plants, and thus herders could prevent livestock fatalities by avoiding these areas. This dendrogram depicts beta diversity using a Bray-Curtis dissimilarity matrix, where 0 implies the same composition is shared between the sites and 1 implies that the sites share no species. From the Bray-Curtis dissimilarity results, it becomes apparent that sites dominated by Tylicodon wallichii and Euphorbia moritanica cluster together at a range of highly similar to the intermediate dissimilarity level, which is between 0.2 to 0.6. Similarly, between sites dominated by Tylicodon wallichii and Euphorbia moritanica may be due to the fact that they were the most prevalent and that they are similar in growth form, both falling under poisonous large succulents. The size coupled with frequency in landscape of these large poisonous plants may then explain why these plants act as a deterrent to herbivores on poisonous plant patches, and ultimately drive species diversity by acting as nurse plants. Furthermore, the similarity in growth form and its poison status might have resulted in these species modifying their environment in similar ways, thus resulting in similar community structures. Thus, sites dominated by Tylochodon vallici and Euphorbia mauritanica cluster together at varying similarity as a result to their unpalatability, prevalence, and similarity in growth forms, which may drive the community structure in similar ways, resulting in similarities between sites dominated by either. With reference to the previous slides, we also found that these species is highly associated with a Coles index of 0.56, which may also explain the similarity. In conclusion, the results of this study provide evidence that poisonous plant patches promote species and growth form diversity, possibly due to deterring grazers. It also sheds light on which palatable plants are associated with poisonous plants in the Steenkopf communal area. This study provides evidence that the ethnobotanical knowledge of herders and the palate of livestock are also major contributors to the spatial distribution of and diversity of plant species in the arid biodiversity hotspot. Thank you very much.